Thank you for the introduction. I will begin by sharing the power of Biotechne with you. Biotechne is a global developer, manufacturer, and supplier of high quality reagents, analytical instruments, and precision diagnostics. Biotechne is a powerful solutions provider with over 750,000 quality products, more than 3,000 committed employees, and now 50 years of innovation experience that can be attributed by over 500,000 user citations. We also offer 7 innovative instrumentation platforms, and one of them, Simple Western, will be the focus of today's talk. And with that, let's get into our Simple Western portfolio. Western blotting and ELISA are established and accepted technologies for protein analysis cited in thousands of publications. Simple Western combines the specificity and size resolution provided by Western blots with reproducible quantitation typically seen with ELISAs. By bringing the benefits of immunoassays and Western blotting together, Simple Western will enable researchers to do things they couldn't do before, such as enabling quantification of protein isoforms and size variants that can now be separated by size to resolve the different protein forms before immunodetection, enabling enhanced detection specificity in complex matrices by leveraging size separation to resolve nonspecific binding and quantify only the target protein at the correct molecular weight, and speeding up method development compared to custom ELISA development timescales, since you now no longer have to waste time and money searching for a suitable antibody pair. What people love about Western blots is that it's easy to create your own assay, but it's only qualitative. What people like about ELISA is that it's shown to be highly quantitative, but it can be challenging to develop, especially in complex matrices. Our vision is to bring you the best of both worlds. Simple Western technology is fundamentally enabled by capillaries. The instruments start by automatically loading the separation matrices into the capillaries, and this is followed by loading a small sample plug in each capillary. Proteins are then separated by their apparent molecular weight, like SDS page. And the next step, which is crucial to this process, is the proteins are immobilized to the capillary wall using UV light. This allows for the removal of the inert separation materials from the capillary. Your Simple Western instrument then performs an immunoassay inside each capillary using the primary antibody of your choice and are ready to use secondary antibodies for easy and sensitive detection of your target. The instrument images the signal from the immunoassay in each capillary and automatically analyzes the data in our Compass software. Simple Western delivers precise, quantitative results in under 3 hours with minimal hands-on time using as little as 3 microliters of sample per well. It is highly reproducible and flexible with exceptional sensitivity while eliminating the need for antibody pairs to streamline assay development. Overall, it provides fast and reliable data across a wide range of samples and targets when you need it. With Simple Western, we offer 3 platforms tailored to your needs. Abby and Jess provide chemiluminescent or chemiluminescence plus fluorescence detection, each capable of running 25 samples in about 3 hours. The Leo platform expands this further, enabling runs of up to 100 samples within the same 3-hour time frame, supporting higher throughput workflows without extending assay time. With that overview, I will pass it over to Mark to now highlight how this technology is applied to analyzing antibody target affinity and specificity.
Thanks for the intro, Madhura. At Roche Tissue Diagnostics, we develop tissue-based cancer diagnostic assays. Many of our test kits rely on monoclonal antibodies that bind to specific cancer biomarkers. To make sure that these kits work, we have to determine that the antibodies bind with high affinity and high specificity. Today, I'll show you how we use Simple Western in our process to do exactly that. Our work process requires us to demonstrate that an antibody being developed for diagnostic use is specific for its target and can detect a broad range of target protein expression. This entails running a Western blot of several cell lines that express high, intermediate, low, or no amounts of the target. The Simple Western system enables us to really rapidly identify cell lines just for this purpose. Here's an example of a Simple Western generated lane view showing cell lines that express a broad range of the target protein that we were interested in. The lanes are blue as I had run this Simple Western assay in the total protein mode. We can clearly see the target precursor, the mature target, and putative breakdown products. On the left is the quantitative electropherogram of the data that was automatically captured during the run without the need for a separate imager as required for a standard Western blot. When compared to the traditional Western blot shown here on the right, it's clear that Simple Western perfectly matches the results using the exact same samples and is a much faster way to achieve this. When developing a diagnostic immunohistochemistry assay, it's necessary to show that the antibody is specific for its target on a tissue sample. We do this using a peptide inhibition assay, where we premix the antibody with a peptide containing the antibody's epitope. At sufficiently high concentrations, the peptide binds all the antibody and then competitively blocks the antibody from binding to the tissue target, which is indicative then of target tissue specificity for that antibody. Let's go over this assay in a little detail. First, the controls. Panel 1 on the left is our negative control. We use a nonspecific antibody that doesn't recognize the target. This results in a light blue stain, which confirms no target binding. Panel 2 uses our specific antibody at full strength, which shows a dark brown stain in the cancerous areas of the tissue. Panels 3 and 4 use the antibody at half strength. Because our inhibition assay dilutes the antibody by half, we need to ensure that this dilution doesn't weaken the signal. As you can see, the stain remains just as strong even when we introduced a random nonspecific peptide as shown in panel 3. Next, we have the actual inhibition assay. Here we pre-incubate the antibody with 5,550 and 5 nanomolar peptide concentrations. At the higher 3 peptide concentrations, binding of the antibody is blocked, but then there is an abrupt transition at 5 nanomolar where full staining is restored. Sometimes we may want an alternative technique to corroborate the results of an IHC and peptide inhibition assay. Simple Western provides an excellent alternative assay for this purpose. In this next panel, we use Simple Western to run the exact same set of controls against a cell line lysate that expresses the target. We see the same result: no staining in the negative control antibody and full staining of equal intensity with the other 3 controls. Just like in the IHC assay, there is an abrupt transition between the 2 lower peptide concentrations that we saw in the IHC assay. Thus, the Simple Western peptide inhibition assay exactly recapitulates what we see with the IHC assay. Next, we can use the Simple Western inhibition assay over a much narrower range of peptide concentrations to further assess antibody binding affinities as shown on this last panel. We've run the peptide inhibition assay at peptides ranging between concentrations of 3 down to 2 nanomolar. On the next slide, we can then further analyze such data. In this slide, I'm going to show you how we can use Simple Western to do a much more quantitative peptide inhibition assay. I've taken the data from the prior slide, where we titrated going from 13 nanomolar down to 2 nanomolar peptide. And as we do that, we get increasing signal strength as we reduce the concentration, a maximum of about 75,000. So we can estimate crudely about a 7 nanomolar KD or IC50 for this run. We can take the data then and put it into a graphing program where we get now a nice sigmoidal shaped curve, and then use the curve fitting aspect of that to generate an EC50 of 7 nanomolar that correlates with our crudely estimated 7 nanomolar value. We're going to use this quantitative peptide inhibition assay in later slides to do additional analyses of the interaction of our monoclonals with their targets. One of the other things we use Simple Western for is to compare the binding affinities of different antibody clones. And in this example, I've taken a rabbit monoclonal antibody and titrated in increasing amounts of the antibody against a fixed amount of the target antigen in the capillary walls. In the other panel, I've repeated the experiment with a mouse clone. We can easily see that the mouse does not bind as well as the rabbit does, but this isn't always the case that you can just visually distinguish between the two. So we'd like to have some quantitative aspect about this. So we can take the data then for the rabbit and put that into a graphing program and then determine that we get a Bmax of about 600,000 counts and a KD of about 10 to 11 nanomolar. If we do the same thing for the mouse clone, what we see now is we get a Bmax of about, 25,000 counts, 27,000 counts, but curiously a KD of 3.5 nanomolar, suggesting that it might be a better binder based on KD, but we can see clearly that by the time it goes through the assay, it's not. So you have to remember the KD is the ratio of the off rate over the on rate, and so you can have different off rates where you might have a low KD, but the thing dissociates so fast that it's going to affect your Bmax as it's done here. And if we overlay the two, we can clearly see there's a 20-fold difference in the Bmax between these two monoclonal antibodies. All right, this next set of slides is going to demonstrate how we use Simple Western's peptide inhibition assay to map exactly where an antibody's epitope is located on the original immunogen. The sequence shown in red on this slide is the full-length peptide immunogen that was used to create the rabbit and mouse clones from our last slide. Here you can see that pre-incubating either antibody with 5 micromolar concentration of the immunogen completely blocks the ability of the antibodies to bind the immobilized target on those capillaries. Again, we can see that the mouse signal is somewhat smaller than with the rabbit. Now, with epitope mapping, what we do is to take the immunogen and break it down into smaller fragments and then test each fragment for the ability to block binding of the antibody. If the subfragment blocks binding, we can assume then that the epitope is contained in that subfragment. If it doesn't block binding, then we know that fragment of the immunogen does not contain the epitope. All right, now, so on this next slide, we're going to take the full-length 30 amino acid immunogen that we saw in the prior slide and do a little peptide inhibition assay at varying concentrations. We start with 5 times 10 to the minus 6, minus 7, minus 8, and minus 9 concentrations. All right, these three higher concentrations block binding. Finally, when we get down to 5 to the minus 9, as we showed before, signal begins to return. So we're going to use the minus 6 and minus 7 molar concentrations in the next set of panels to see whether or not subfragments of this full-length immunogen have the ability to block binding of the antibody to the immobilized target on the capillary walls. So now if we take the amino-terminal third of the immunogen, we can see that minus 6 and minus 7 concentrations really block it quite effectively from being able to bind. So we assume then that the epitope is within this amino-terminal third region. If we go to the central region of the immunogen or to the the carboxy-terminal third of the immunogen, we don't see any blocking of signal. So it must be in this region here. If we repeat this now with the rabbit antibody, what we see is, once again, the higher concentrations block it, as we expect. And then if you look at the amino-terminal third of it, that region still blocks, but then the central region and the carboxy-terminal third of the immunogen fail to block binding. So for both antibodies then, the amino-terminal third of this immunogen probably contains the epitope. Okay, on the prior slide, we showed that the N-terminal third of the immunogen probably contains the epitope of both of these antibodies. So what we're going to do then is subfragment this into smaller fragments and then assess the ability of a 5 times 10 to the minus 7 molar concentration of each of the peptides to block binding of the antibody to the capillary walls. If it blocks binding, then we know that particular subfragment probably contains the epitope for that particular antibody. All right, so here we're going to start now with a smaller fragment of the N-terminal region. And what we see is that neither antibody is blocked in its ability to bind. So this extreme terminal region doesn't contain the epitope. Now, if we slide over a few more amino acids and look at the central region of this N-terminal fragment, we now see that the mouse antibody is blocked from binding, so the mouse antibody probably resides in this fragment, but the rabbit antibody still binds, so its epitope cannot be in this fragment. And now if we slide over a few amino acids more towards the C terminus of this domain, we see now that we've lost binding of the mouse antibody, but the rabbit antibody is blocked from binding, so the rabbit epitope probably resides in this region. So now I'm going to show you kind of a summary of this that's a little easier to follow on the next slide. Okay, so in this slide, we're going to show then a little table that summarizes the data that we just saw, where we mapped the probable location of the epitope for each of these two antibodies. We started with the full-length imaging, and we showed that it could block binding of both of these antibodies to the immobilized target on a wall. So that means the antibodies bind to the imaging quite well. Then, if we take the C-terminal half of that imaging, we found they couldn't really block binding of the antibody to the capillary walls. Therefore, the antibodies do not bind in this region, and the epitope is not to be found in that region of the imaging. Right? We next took the central third of the imaging, and the same thing. We found that this failed to block binding of the antibodies, therefore the epitope cannot reside within this region. So then we went over and took the amino terminal half of the imaging and found that that blocked binding of both antibodies to the capillary walls. Therefore, we knew that the epitope for each antibody has to reside somewhere within this amino terminal half of the imaging. All right. So then if we take out several amino acids to the C terminus of that and ask, does this fragment block binding of each antibody to the capillary walls? The answer was no. Therefore, this does not contain the epitope for either antibody. And then if we slid over a few amino acids to the right of this N-terminal fragment, we found in this case, the mouse binds, but the rabbit does not. And then sliding over a few more amino acids to the right, we find now that the mouse no longer binds, but the rabbit does. And therefore, these two guys do share some amino acids in common with their two epitopes. So now I'm going to show you on the next slide for the rabbit antibody is we're going to do some really fine mapping with that. So we're going to use peptide inhibition along with alanine scanning to further refine where the critical amino acids are for binding for the rabbit epitope. Okay, on this slide, I'm going to show you how we use the simple Western peptide inhibition assay described in the previous slide with two other orthologous techniques that will also utilize alanine substitution to measure the contribution of each amino acid in the epitope towards binding. All right, so we see here then is where I've done a simple Western peptide inhibition assay on the wild type sequence and then derived an IC50 with that. We're going to repeat it, and we're going to have an alanine substitution along each position along this sequence and measure the binding of each of those in terms of an IC50. Then our second assay uses surface plasmon resonance. In this technique, polarized light reflects off a chip containing an immobilized antibody. The reflection angle changes based on the mass at the surface, which is determined by the protein that's bound on the surface. When we flow the peptide over the chip, it binds to the antibody and then increases the mass on the chip, thereby altering the angle of light reflection. Measuring this shift in the reflection angle over time gives us the on rate, or a Ka. When we then flow buffer over the chip to monitor the target dissociating, we'll get an off rate, or a dissociation constant. From these two rates, we can then calculate the overall dissociation constant and the half-life of the antibody peptide complex for every alanine substitution along the epitope. Finally, what we're going to use is just our simple IHC assay on a tissue slice, where we're going to do an abbreviated peptide inhibition assay with 5 micromolar, 0.5 micromolar, and 0.05 micromolar. And then we're going to score that as a 0, a 1, a 2, or a 3. And then we'll also determine the percentage of cells that show a positive stain. All right, now I'm going to show on the next slide examples of the types of effects that we're going to see with these alanine substitutions. Okay, now I'm going to show you the three possible outcomes when combining these three separate techniques of alanine scanning mutagenesis to assess the contribution of each amino acid to binding to the antibody. The first is going to be no effect. The substituted amino acid is not involved in binding, so the simple Western that the SPR results and the IHC results are all going to be identical to what we see with the wild type sequence. In the second one, we'll see a moderate effect. The amino acid contributes to binding, but it's not really essential for binding. In this case, the simple Western curve will shift to the right. The SPR signal shows a slightly reduced maximum response, but a faster dissociation rate, and the IHC requires a higher peptide concentration to achieve inhibition. Then finally, there's a major or catastrophic effect. The amino acid that's substituted turns out to be critical for binding. The simple Western curve shifts to the right by a couple of orders of magnitude. The SPR detects no binding event, and the peptide completely fails to inhibit binding in the IHC assay. Okay, on this data, we're showing a table then where we've taken the alanine scanning data using all three techniques and combined them into this one table. Here is our original wild type epitope sequence. I got an IC50 by simple Western of 11 nanomolar. KD is lower by SPR, about 0.3 nanomolar. 211-minute half-life, pretty stable complex. And then you have to drop all the way down to 50 nanomolar to see the signal beginning to return by IHC. All right, here we have a couple of catastrophic results. When we substituted this histidine or this leucine, we lost binding pretty much altogether. IHC, now the IC50 goes up to 3,600 nanomolar, 6,000 nanomolar. Don't even see binding by the SPR technique, and you really have to add, you can't even see it when you add 5 micromolar of the peptide. And then here are a few examples of we had an intermediate effect, like right here where we substituted this proline with an alanine, see an increase from 0.3 to 6.9. Half-life now is only 5 minutes. And then there are a number that really didn't have any effect, like out here towards the ends on either side. All right, now I'm going to show you a graphical summation of this result on the next slide. All right, so what I've done on this next slide is to take the data table from the previous slide and rearrange it so that now it's visually easier to assess the contribution of each of those amino acids in the minimal epitope that we've identified towards binding to the antibody. Amino acids in black are not essential for binding. Those in yellow contribute to binding. Those in red are critical for binding. And the blue one, we don't know because it was originally an alanine in the epitope itself. All right, so as we march across the board there, that first proline is not required for binding. The glycine is contributory. The histidine here is essential, proline contributory, leucine critical for binding, alanine don't know, proline contributory. This is one where I didn't quite see agreement between the three techniques. The simple Western didn't show that this fellow here, this glycine, played a contributory role, but the SPR and IHC suggested that it does play a contributory role. But then the next three amino acids, according to all three techniques, just weren't critical for binding. So then we've gone from an 11 amino acid sequence that we've narrowed down as our epitope to identifying the critical amino acids in that epitope as being a 7 amino acid stretch. So out of the original 30 amino acid Imugen, we've now got a 7 amino acid stretch. And now we can use this real minimal epitope to go through BLAST-P and use that information to search for other proteins that could be potential off targets. All right, now I'm going to show you next how we can use simple Western combined with other techniques to identify off targets of binding. Now let's see how simple Western can help track down off-target staining using the HER2-specific antibody 4B5 as an example. In the three different tumor tissue sections on the left and BC3C bladder cancer cells on the right, 4B5 shows strong cytosolic staining when it should mostly just stain the cellular membranes. Staining these same samples with a highly specific HER2 control antibody shows almost nothing, with the exception of a little bit of membrane staining on the upper tissue sample. This likely rules out cytoplasmic HER2 fragments as the source of the stain. We also know that 4B5 cross-reacts with HER4 and Z-scan 18. But mRNA expression levels of HER2, HER4, and Z-scan in this BC3C cell line are just simply way too low to see by IHC staining. So we can likely rule out these known off-targets as the cause of the unexpected staining. Okay, to further investigate the source of the 4B5 off-target staining that we observed in the prior slide, we ran a series of traditional Western blots using cell lysates from several different cell lines and probed those blots with either the 4B5 antibody or with the comparator antibody. And what we're seeing in this panel is a blot that's been probed with 4B5. The first lane has the BC3C cell line lysate that we discussed from the prior slide. And what we see is the presence of a single 37 kilodalton band. In the next lane is a different cell line that expresses a high amount of the HER2 protein. We observe the presence of the full-length protein up here as a 200 kilodalton band and the presence of a 45 kilodalton known breakdown product, but we do not see any of this 37 kilodalton band. We also observe this 37 kilodalton band and at higher levels in several other cell lines shown here. Right. So in the next panel, what we did is to fractionate one of these cell lines, the A549, into the cytosolic fraction, the membrane fraction, the nuclear fraction, and the insoluble. What we observed is this 37 kilodalton band primarily occurs in the cytosolic fraction, with moderate to small amounts in the membrane fraction, with the full-length HER2 up here, but with only traces of it occurring in the nucleus and in the insoluble fraction. So then we repeated this experiment, but then this time we probed with the comparator antibody. And what we observe is the expected full-length protein in the membrane fraction, but not in the cytosol, and only traces in the nucleus when compared with the full-length purified recombinant control protein HER2, but none of the 37 kilodalton band. So what this tells us then is that the 37 kilodalton protein is either an off-target unique to 4B5, or it could be a HER2 fragment that is just simply missing the epitope of the comparator antibody. So let's now investigate this further on the next slide. Okay, to identify this mystery 37 kilodalton protein, the team took cytosolic fractions from several cell lines and subjected them to ion exchange chromatography. We then used simple Western with replex to locate the fractions that had the 37 kilodalton protein. So what we see here is a fraction that expresses the most amount of the 37 kilodalton protein, but it also has, as determined by replex, a fair amount of contaminating protein, which the adjacent fraction does not. So even though the adjacent fraction has a little less of the 37 kilodalton protein, there's substantially less contaminating protein in that fraction. We then subjected these fractions to immunoprecipitation as shown in this panel. All right, so here we have several different cell lines that we've immunoprecipitated. Here's our starting input material here. Here's the 37 kilodalton band from 3 cell lines, and our NCI N87 control cell line, which expresses the full-length protein, but not the 37 kilodalton band as shown here. And if we immunoprecipitate all of these guys, we now see that in the A549 cell line, the 37 kilodalton band is being brought down nicely, not so much in these other 2 cell lines. And in our control cell line, we're bringing down the full-length HER2 protein, but none of that 37 kilodalton band. So now what we do next is to subject this band to mass spec by excising the band and sending off to our mass spec facility. And what the facility identified was AKR1B10 as a probable cross-reactive protein with the 4B5 protein. So now let's investigate that a little bit further on the next slide. Now, on this slide, we're going to use Simple Western to demonstrate off-target binding of the HER2 4B5 antibody to the AKR1B10 protein that was identified by mass spectrometry. The leftward two capillaries contain our positive recombinant HER2 control that we probed with 4B5. The next two capillaries contain recombinant AKR1B10. We see a faint signal, but it's very definite, of binding 4B5 to this recombinant protein. Then the next four capillaries contain cell lysates of A549 and the BCC3 cell lines, where we get a strong signal that lines up with the AKR1B10 signal. Then let's take it to the next step, where we now repeat the experiment, but instead we now use anti-AKR1B10 antibody. This antibody binds strongly to the recombinant AKR1B10 protein in these lanes, and it also gives a signal comparable to what we see in the 4B5 stained capillaries with the A549 and BCC3 cell lines. This result strongly suggests then that the AKR1B10 is indeed a new cross-reactive protein for the 4B5 antibody. Okay, we've determined that AKR1B10 is a likely off-target for binding to the 4B5 antibody. But where in AKR1B10 is the likely epitope? Well, we can glean that from here. I've taken the HER2 protein and aligned the epitope that binds to 4B5 with three other proteins that bear sequence homology to the epitope. One of these proteins does not bind, and we've determined that this proline is essential. These other two proteins bind with different efficiencies, HER4 pretty well, and the other Z-scan one not so good. So we went looking then for this minimal motif that falls out of this, of being a glutamic acid, a proline, and a leucine, and asked, does the AKR1B10 have that motif? Sure enough, it does. So now what we can do is synthesize this peptide and see if it works in a peptide inhibition assay on the next slide. So, we next performed a 4B5 peptide inhibition assay against HER2 protein using three different peptides at high and low concentrations. As expected over here on the left, the HER2 epitope peptide completely blocks 4B5 binding at the high concentration, while a random control peptide showed no inhibition at all. Next, an AKR1B10 peptide containing the minimal 4B5 binding motif shows substantial inhibition, confirming that AKR1 does cross-react with 4B5. Comparison of the peak areas of the high peptide concentrations, however, shows that the AKR1B peptide is about 32-fold less effective at inhibiting 4B5 than the HER2 epitope. On the next slide, we will use Simple Western to generate data that more quantitatively shows the relative affinities of the 4B5 antibody for its intended HER2 target and three off-targets. Okay, on this slide, we're going to use Simple Western to do comparative quantitation of 4B5 off-target binding against recombinant HER2, HER4, Z-scan 18, and our newly discovered AKR1B10 protein. The experiments are done by titrating increasing amounts of each recombinant protein against a constant concentration of the 4B5 antibody. We can see that the signal increases across the board as a function of the concentration of all four proteins. We can also see the off-target proteins required higher protein concentrations to approach saturation. If we now pop this data into our graphing program and overlay the plots where we display the percentage of maximum luminescent peak area versus the log of the protein concentration, we can see that the off-target binding curves are shifted to the right, beginning with HER4, then next over is Z-scan 18, and finally with the newly discovered AKR B110 off-target. If we now show the quantitative results of these in terms of EC50s, we can see the HER2 has about 11 nanomolar EC50. Compare this with H4, which is 32 nanomolar, or about threefold less efficient, Z-scan 18 with a EC50 of about 200 nanomolar, and then finally the AKR1B10, which is 625 nanomolar, or approximately 60-fold less efficient binding than with the HER2 protein. So we suspect that the visible AKR1B10 cross-reactivity in tissues occurs because multiple AKR1 family proteins are expressed simultaneously at high concentrations in certain tissues. So ultimately, quantifying these off-target interactions enables us to adjust assay conditions in diagnostic assay kits and to develop algorithms to mitigate any diagnostic interference that these off-targets may have. So with that, now I want to summarize on the last slide what we've learned today. Okay, this last slide just summarizes then all the things that we can use Simple Western for in analyzing monoclonal antibody target specificities and affinities. We can use Simple Western to rapidly identify panels of cells that express high, intermediate, and low levels of the target. We can use peptide inhibition assay of Simple Western to corroborate IHC peptide inhibition assay results. We can also use Simple Western then to estimate and compare antibody clone binding affinities. We can use Simple Western to identify epitope sequences via peptide inhibition assays on fragments of sequences containing the epitope. Then we can do fine mapping of those sequences to identify critical residues that are essential for binding, again using peptide inhibition assay analyses. Finally, we can identify, in conjunction with other techniques, monoclonal antibody off targets and then quantitate the relative affinity of those off targets with that of the actual target itself. And with that, I thank you very much for listening.